Okay, this is going to be Chapter 8, the nervous system. And this is going to be Part 1. There's going to be 3 parts to this chapter. So, introduction here. Central nervous system versus the peripheral nervous system. CNS versus the PNS. The central nervous system is essentially the brain and spinal column. Not drawn to size or scale. Brain, spinal column here. And the peripheral nervous system is responsible for the input. So whenever we look at this, we have the peripheral nervous system out here that is either controlling the organs or getting information coming in sensory or sending information to do a specific task, and the central nervous system is essentially running the entire operation. Soma versus axon, and we're talking about a nerve cell. Right here will be our soma body. Axon, dendrites. So, connect the anatomy and physiology. Dendrites and the soma gather the information, so that's this guy up here, pretty much the cell body. And the axon conducts the action potentials or the actual stimulus, or the input, either way. So this could be receiving input, or this could be receiving output. I need you to do a specific task. Afferent versus efferent. Afferent pretty much means towards the CNS, and efferent means out of the CNS or exit. E for exit, A for towards. And we'll look at this later. It'll make a little bit more sense here in just a moment. Sensory versus motor. In contrast to afferent versus efferent, or out, or toward, my apologies, in or out of the body. These terms indicate the function of an axon. So either you're going to get sensory input in to the brain, essentially, or you're going to get motor output. Dorsal root versus ventral root. The dorsal is strictly sensory, and the ventral is strictly motor. So on the dorsal horn, what we're going to see is we're going to see the input, or the sensory input. Is it hot? Is it cold? What's the texture of this? So that's all would be sensory input. And then the ventral portion is going to be motor. It is hot. Please move your arm. That would be a reflexive action. Dorsal is sensory, and ventral is motor. Receptor versus effectors. Organs of the peripheral nervous system will either receive information or send it to the central nervous system, or will receive stimuli from the central nervous system and take some action. So what this means is the peripheral nervous system is set up to where, let's say that we have the liver, not drawn to scale. The peripheral nervous system would have autonomic input and autonomic sensory to look at, see how well this organ is doing, and maybe activate the endocrine system if required or necessary. The CNS will receive stimulus and take some action. So all of this sensory input, whether it be autonomic or sensory that we can actually feel, will be headed towards the central nervous system, and actions will be taken. Generally, whenever we're cascading or telling multiple organ systems to interact, this would be facilitated by the endocrine system. Visceral versus somatic. In general, visceral is associated with involuntary or automatic control. You can also think organs whenever we're looking at this. You have no physical control over the amount of glucose that's released from glycogen stores in your liver. The visceral portion of this would be that this is run automatically or autonomically in your body. The somatic version of this is associated with voluntary control. So this would be like picking up something like a Coke bottle or a pen or something like that, that you had physical control over it. Gray matter versus white matter. Associate gray matter with cell bodies. So whenever we talk about cellular bodies, somal bodies, and dendrites, and white matter with the myelin sheaths, or myelin, and this is going to be down the axon ends. And we're going to learn terms here later on to where these things are going to be myelinated. Now what this allows is for speed and efficiency as far as action potential goes, or the electrical movement in the axonal body. When it's myelinated, it is allowed to essentially pawn skip over these gap junctions here. The action potential keeps going until the information, but to speed and enhance the responsiveness, that's what the myelin sheath is for. Input versus output. So on the input side, and this is going towards the brain, so I'm going to draw a brain here again. Poor brain drawing, I know, but you get the picture with this. So for input side, we have afferent, sensory, or dorsal. So this is all going to be going into the central nervous system. More specifically, I drew it into the brain. It would go down into the spinal column for sensory input. And then we have output, which is known as efferent, which would be motor response. This would be moving objects. This is going to be on the ventral side. And this is going to be our actual motor senses, like move a pen, walk, take the hat off of our head, and things like that. So output. 2 organ systems. And this actually starts the slide set here. The rest of that was just to try to, as we start talking about specific terms, to try to keep them square in your head. So you may have to review that first portion a couple of times. 1.0, two organ systems, and the 2 organ systems we're going to talk about here are the nervous system and the endocrine system. And they're pretty much an integral part of one another. The endocrine system pretty much coordinates the organs in the body to maintain homeostasis, and the nervous system pretty much quickly responds to a changing environment. So this has control over motor and the autonomic nervous system, or the ANS. And what this will allow us to do is, without forethought or out any kind of thinking, interact with our environment. So if it's too cold, the body starts shivering without our conscious thought. If we need more sugar stores, the body will adjust this accordingly, and so on. So the 2 organ systems that are going to make this happen are going to be the endocrine and the nervous system. 2.0, the nervous system. Cellular organization and neural tissue, neuron function, and neuronal communication in this section. So 2.1 here, the nervous system, pretty much does 3 things. It monitors the internal and external environment, it integrates sensory information, and it coordinates voluntary responses in organ systems. This is all controlled by cells called neurons. And the central nervous system pretty much consists of the brain and spinal cord. The peripheral nervous system, all neural tissue outside of the CNS. So anything outside of the brain or spinal column is considered peripheral nervous system. And there are 2 divisions of this peripheral nervous system. There is afferent, which means towards. So brain here, afferent would be towards. And there's efferent, and this would be out towards the muscles, or out of, or exit. Somatic nervous system, control over skeletal muscles and contractions. That we have physical control over the somatic nervous system. And then we have the autonomic nervous system. And the autonomic nervous system provides automatic involuntary regulation of smooth muscle, cardiac muscle, glandular secretions, adipose tissue. The autonomic nervous system has 2 subsections, and if you'll start looking at this like a teeter-totter, it'll make a little bit more sense. We have the sympathetic and the parasympathetic nervous systems in the autonomic nervous system, and it works exactly like a teeter-totter. And each one of these do kind of the opposite of the other. The sympathetic nervous system is fight or flight. It would accelerate the heart or get us ready to go toe-to-toe if we saw something that was threatening. So we would get an increase in the sympathetic nervous system, or adrenaline, or fight or flight. And then we have the parasympathetic nervous system. This, like, feed or breed or rest and digest is what it's termed with. So it would do the opposite calm things, decrease catecholamines, digest food better. So both of these nervous systems respond just like a teeter-totter. So if you have an increase in sympathetic tone, you're going to have a decreased parasympathetic tone. If you have an increased parasympathetic tone, you have a decreased sympathetic tone. And they maintain homeostasis in the body. 2.2 divisions of the central nervous system, and we have the central nervous system kind of up top here, and it does information processing. And then we have the peripheral nervous system. Now this is going to be outside the spinal column and outside the brain and spinal cord. And in this we have two divisions. We have a sensory afferent division, which is going to give us information to the CNS. And then we have an afferent division. Now one of these we have control over, which is somatic, and this is going to be skeletal muscles that we can move things with. And the other one is going to be the autonomic nervous system, which we don't have control over. The autonomic nervous system is automatic. So in that autonomic nervous system we have two more subdivisions, and these are the ones we just got through talking about, which is the sympathetic and the parasympathetic nervous system. And these work on smooth muscle, cardiac muscle, glands, and adipose tissue. So all of this stuff here that we would secrete, specific hormones or specific types of neurotransmitter, are all going to be done without conscious thought. It's going to be done automatically. In going back over here to the afferent side, we have receptors that pretty much receive information, somatic sensory receptors, and then we have visceral sensory receptors. The somatic version is pretty much touch. Is it hot? Is it cold? What does the texture feel like? So it gives a sensory input on any event. And then we have visceral sensory receptors, which monitors internal conditions like our organ systems, liver, heart, lungs, and so on. 2.3, cellular organization in neural tissue. And in this we're going to talk about, and there's lots of terms in this, neurons, structural classifications of neurons, functional classifications of neurons, neuroglia, demyelination disorders, and anatomical organization of the neurons. 2.4, cellular organization in neural tissue. We have the neurons, which are pretty much the basic unit of the nervous system. So whenever we're talking about the actual nerves themselves, they are essentially neurons, and this would be the nerve cell. And then we have neuroglia, and these regulate the environment around the neuron. And if we want to think of neuroglia as pretty much a pit crew that allow the neurons to function in their environment, then that would be a pretty good thought process on the neuroglia. 2.5, this is going to break down the neurons a little bit farther. A neuron has four parts: a cell body, branches that come off of it called dendrites, and this is going to be for incoming information, an elongated axon, and this is going to be for outgoing information, and then synaptic terminals, and this allows communication with other cells. And again, here we go. This is the somal body, or the cell body. These up here would be the dendrites. These are going to accept information from other things. This is going to be the output side, where information is being output on, and this is the axon. And if there was another axon like this had to talk to multiple organ systems at once, this would be a collateral. This may be talking to skeletal muscle and then to tendon simultaneously for actual movement. So again, information input through the dendrites communicates with the somal body. The somal body responds in an action potential that runs down the axon and will tell supporting structures or the actual target organs what to do. Structural classification of neurons. Multipolar, which means two or more dendrites common, controls pretty much skeletal muscles. We have unipolar. Dendrites and axons are continuous and the cell body lies off to the side, and these are different types. We'll show these here in just a second. Lies off to the side. Sensory neurons of the peripheral nervous system are an example of these unipolar neurons. Bipolar neurons, which have one dendrite and one axon with the cell body in between, rare but occur in special sense organs. So this is a multipolar neuron up top here. This is a unipolar neuron, and this is where it gets its information from. And then this is a bipolar neuron, which has both ways. Now this one here would communicate with a very large amount of neurons that may be in conjunction here, and more dendrites off of those, axonal bodies, synaptic clefts on the end, and so on. Functional classifications of neurons. We have sensory neurons, and these would be the afferent division of the peripheral nervous system, where sensory neurons would come from. Somatic sensory receptors, outside world information. So these are going to be external receptors, information about the external environment. Is it cold? Is it hot? How does it feel? And then proprioceptors, which monitor position and movement. So I am standing upright, or I am laying flat on my back. So these are all specific types of receptors that we see from the sensory neurons afferent division, and this is going to be information put into the central nervous system from the peripheral nervous system. And then we have visceral receptors. In this, visceral receptors would monitor the activities of the digestive, respiratory, cardio, urinary, things that we wouldn't have conscious thought over. So input into the central nervous system, how well are the organs doing? Do I need assistance from another organ system, and so on. Motor neurons carry out instructions from the central nervous system, and these can be somatic and visceral. Somatic would be innervated skeletal muscles. So move your arm. This would be coming down the somatic side. And then also we have visceral, which pretty much means organs, viscera, visceral organs. Innervated effector organs, cardiac muscle or smooth muscle glands within the actual organs themselves. And then also in this we have interneurons, brain and spinal cord, and these interconnect neurons. So these allow for collateral communication between the neurons in larger structures. And an example of this is going to be the brain and the spinal cord, where it's coming into the central nervous system. 2.10 neuroglia. And these are found in the central nervous system and the peripheral nervous system, but a greater variety of glial cells are in the central nervous system, and this would be the brain and spinal cord. We have various types of these, which are, one of them are called astrocytes. They pretty much, and these are the supportive structures for the neuronal environment. They secrete chemicals vital to the blood-brain barrier and create structural framework for the brain. Oligodendrocytes, sheaths and insulates around axons, and we have a couple versions of this, which are myelin, is a term here. Main purpose of a myelin layer or sheath is to increase the speed at which the impulses propagate along a myelinated fiber, and this is what I termed earlier as pond skipping. So if you have an axonal body, I'll draw one up here, and it is myelinated, which means it has like a protective structure around the actual axon. The speed at which the action potential comes out could be increased due to the pond skipping effect. So action potential is traveling, but to get the stimulus out ahead so that the stimulus can actually occur in the axon, pond skipping occurs. So now your action potential is traveling at a much faster rate. Myelinated axons are coated with myelin, and then we have unmyelinated axons, and these are pretty much without myelin. Microglia, the smallest and rarest, and they're phagocytic, which means that they remove foreign materials and pretty much eat other cells that don't belong there. Ependymal line both the central canal of the spinal column and the chambers of the brain, and then we have amygdala. In some regions produce cerebral spinal fluid. Cilia on the cells help circulate the fluid within the CSF. So these things are going to produce or assist with our environments. Ependymal cells, some of them, amygdala, actually would make CSF and produce CSF, and will move it throughout the body through little cilia or finger-like projections that they'll have. Neuroglia in the peripheral nervous system, we have a couple different types. We have satellite cells, which surround and support neuron cells, bodies in the peripheral nervous system, like astrocytes do in the central nervous system. And then we have Schwann cells, and they cover every axon outside of the CNS. Outer surface of the Schwann cell is also called neurolemma. Can cover only one segment of a single axon. Can also enclose portions of several different unmyelinated axons. So it's a way of keeping communication the Schwann cells keep the communication going the same direction on something that's unmyelinated, so that we don't get collateral information. You could tell your finger to move and actually get your toe to move accidentally. So a Schwann cell would assist with the direction of the communication. And these are all of these neuroglia. Ependymal cells, which would be here, in the central canal. This is a cross-section of the spinal cord. We have gray matter, which would essentially be our somal bodies. And then we have white matter, which would be the axons, and either they are myelinated or unmyelinated. If they are unmyelinated, that would be this side over here. And this astrocyte is providing structural support for those axonal bodies. And then in the case over here, we have a myelinated version, which allows for pond skipping. And we also have oligodendrocytes that are assisting in that. And then these up here would be the somal bodies, dendrites, axon. So please look over and be able to identify these structures. Figure 8.5 in your book, 2.13 slide, is Schwann cells and peripheral axons. And these are going to be unmyelinated. You have a myelinated version here and an unmyelinated version. The one on this side is unmyelinated, and these are the Schwann cells, which assist in the communication, allow for communication to flow so that you don't get collateral communication on here. And then we have myelinated axons, and these allow for the action potential is going to be coming down the axonal body, and the information is going to jump out ahead of time so that this is nice and clear and concise, so that your actual action potential can travel down that axonal body. Clinical note, demyelination disorder. So the myelinated sheath allows for like an insulated barrier. Progressive destruction of the myelinated sheath in the disorders, accompanied with inflammation and axon damage and scarring of neurotissue. Multiple sclerosis is an example of this. Is a disease in which the fatty myelin sheaths around the axons of the brain and spinal cord are damaged, leading to demyelination and scarring. And Guillain-Barré syndrome, disorder affecting the peripheral nervous system, usually triggered by an acute infectious process. It's caused by an autoimmune response directed against the Schwann cells of the actual cell membranes. And then diphtheria, which is a disease that you can actually get. We generally get immunizations for this whenever we get a tetanus. Diphtheria, pertussis, and tetanus toxoid is generally given our DPT whenever you get a tetanus shot. Or the booster sometimes is isolated, but most of the time we get a DPT injection. And this will cause peripheral neuropathy in the peripheral nervous system. Anatomical organization of the neurons. We have the peripheral nervous system and the central nervous system. And in the peripheral nervous system, we have ganglia, with another term, which are neuron cell bodies in the gray matter. And then we have white matter. In the peripheral nervous system, it contains axon bundles together within nerves. So peripheral nervous system, ganglia, and white matter. In the central nervous system, center is a collection of neural bodies with a common function. And then we have the neural cortex, portion of the brain surface covered by a thick layer of gray matter, and the gray matter was the somal bodies. So these guys are going to be collaterally communicating and making specific functions depending on their location in the brain. Then we have tracts and columns. White matter of the CNS contain bundles of axons. These are called tracts or columns. And then we have pathways, which link centers of the brain with the rest of the body. Sensory pathways, ascending, distribute information from the sensory receptors to processing centers in the brain. And then we have motor pathways, which are descending, begin at the central nervous system centers, concerned with motor activity at the end of the skeletal muscles they control. So we have sensory pathways, which are essentially input, and then motor pathways, which are essentially output. And this is kind of an organization of the nervous system, a picture. Okay, so let's start off with the central nervous system on this side, and then we have the neural cortex, primarily composed of gray matter, and the gray matters would be somal bodies. So now you have all these little axons running down in the white matter, and then again here you have gray matter, so again more somal bodies receiving information, and then again running down. So we're going to see somal body, axon, somal body, axon. Nuclei, collection of neuronal cell bodies in the interior of the brain. You're going to get communication occurring, sensory input in this direction, and communication occurring from the top, conscious thought, down towards autonomic running areas. You have centers, collections for neuronal cell bodies in the central nervous system. Each center has a specific function as far as the neuronal centers go, or the gray matter centers. And then you have higher centers, most complex centers of the brain. So higher brain functions would be composed off of these areas here. White matter organization, which again is primarily composed of axonal bodies. You have tracts, which are bundles of CNS axons, and they share a common origin, destination, and function. Example of this is the cranial nerves would control most of the autonomic function of the primary organs, lungs, heart, intestinal movement, and so on. They also would control motor function, which we'll see that we can actually see deficits of whenever someone has a brain injury or intracranial swelling or some kind of spinal cord injury. On the left side here we have columns, several tracts that form an anatomically distinctive mass. And then we're going to talk about the peripheral nervous system on this side. We have two types of matter still in the brain tissue, which are gray matter and white matter. The gray matter is composed of the ganglia, which are a collection of neuronal cell bodies, again, somal bodies with dendrite access. Then we have white matter, which are nerves, bundles of axons in the peripheral nervous system. Now these can be quite long. An axonal body will go from the point of origin or somal head to the actual effector tissue. This could be as long as your leg, as long as your arm, and so on. They generally will branch off and hit multiple muscle systems simultaneously. So this is how your brain and body communicate with your varying arms, limbs, and organ systems in their body. 2.17 neuronal function, the membrane potential, neurotoxins in seafood, and propagation of an action potential in this section. Membrane potential, we have a polarized membrane, state of readiness, which whenever we say polarized, which means it is ready for an action potential or electricity or a stimulus to travel down it. And then we have the potential difference, the difference between the positive charge on the outside and the negative charge on the inside. Next term is membrane potential, and that's essentially going to be the potential difference, or does this thing have a potential to generate a stimulus. And then last term here is going to be resting potential, the membrane potential of an undisturbed cell. Changes in membrane potential. So how we are going to make a stimulus occur is going to be called depolarization. So depolarization occurs because of the opening of a sodium-potassium channel. The opening of the gated sodium channels that increase the positively charged ions on the inner surface of the cell, and the membrane potential shifts towards zero. Whenever you're at a resting membrane potential, it's towards the negative side. So the more zero it gets, the more polarization we actually see occurring, and it receives a charge. So as this becomes a positive millivolt, what we're going to see is the action potential kind of takes over a life of its own and then will initiate itself. Hyperpolarization is considered overpolarization, negative 70 millivolts to negative 80 millivolts. Graded potentials, change in a potential that cannot spread far from the site. An action potential, and this is the term that's most important in this section. The change in membrane potential of the entire cell. Skeleton and axons of the neurons can generate an action potential. So the action potential is the stimulus itself. We have something here called threshold. Membrane achieves a change of a specific level. And then last but not least, we have another theory that's considered the all or none theory. A given stimulus either triggers an action potential or a movement of electricity, or it does not. It doesn't do a half-rated job here. It won't initiate halfway and then say, oh, there isn't enough. It's either going to occur or it's not going to occur. There's no halfway in between. And then this would be the cellular membranes at a resting potential. And this is figure 8.7 in your book. Millivolt negative 70, if we'll take a look at this. We have gated channels here that start, and the sodium potassium pump is essential. This is how the sodium potassium pump is going to allow us to change the charge on the inside and the outside of the cell. Now whenever it gets closer and closer and closer to zero, we'll get an action potential actually occurring, and the electricity will travel down the membrane. The generation of an action potential. We have something called a refractory period, and this is a period from the moment that the voltage regulated sodium channels open at threshold, which is just before all this starts, until repolarization is complete. The membrane cannot respond to a stimuli, and this is considered the refractory period. So this is depolarization, this is figure 8.8, and this is the generation of an action potential. So step 1, sodium ions are on the outside, and then step 2, activation of the voltage regulated sodium channels and rapid depolarization. So what we're going to see here is we're going to see sodium rapidly moving inside the cell, and potassium, and the cell in here is going to become more and more and more positively charged. Inactivation of sodium channels and activation of voltage regulated potassium channels. So at that point potassium is going to be kicked out, and this is the sodium potassium pump. So whenever we get threshold is achieved, so all of this is in a polarized state ready to go, and then what we get is we get the sodium channels coming in and we achieve something called threshold. Now at the point of threshold, this is where the all or none theory comes in. So either this action potential is going to occur or it isn't. So whenever the sodium potassium pump initiates at that point, we start gaining voltage or gaining a positive charge. Depolarization occurs, or actual contraction in the muscle case, or movement of the electricity in the nerve case, whenever we get a positive, just below a positive 30 millivolts. During this phase here, we have to do it all over again, so this is considered repolarization. And this right here, from the time of initiation to the time that it's ready to get back at a resting potential state, is considered the refractory period. During this time frame, we can't receive any other stimulus. Step 4, returns to a normal permeability in a resting state. Clinical note, neurotoxins in seafood. This is going to be 2.23. Several toxins come from seafood, neurotoxins. Tetrodotoxin from pufferfish, saxitoxin from bivalve shellfish, Sigura toxin from carnivorous fish. An example is this, and the toxins prevent sodium channels from opening. Neurons cannot function in this environment. So these neurotoxins would actually cause no stimulus to occur in your neuronal cells. Propagation of an action potential continued here. We have continuous propagation, and these are small steps, depolarization in front with a refractory period in the back. And this may occur in unmyelinated axons. And then we have a saltatory propagation, and this would be pond skipping on myelinated axons. An example of this, the pond skipping version is going to be quicker and more efficient. So this one here is occurring in unmyelinated axons, and what we get here is the sodium-potassium pump when it initiates, and then our action potential will travel. But this has to go started here and travel all the way down, so the unmyelinated version is a little slower than the myelinated version. This right here is the myelinated version of the action potential. Stimulates depolarized membranes to threshold, and then what occurs is that in between this myelinated, these gaps here, we get the charge pond skipping. So then it brings up the action potential faster to the local current here as shown, and so on down the axon of the myelinated axon. 2.26 neuronal communications, and this occurs through synapses. We're going to talk about structure of synapses, synaptic function in neurotransmitters, organophosphate poisoning, and then neurotransmitters in this section. Structure of a synapse. We have essentially a synaptic knob, head of a presynaptic neuron, a presynaptic neuron, the knob or the end of an axonal body, and then a postsynaptic neuron, and this would be the other receiving neuron if we're going nerve to nerve, or this might be the motor end plate if we're going from nerve to target organ. And then we have a synaptic cleft, and this is a narrow space between the neurons. So this is the axonal body here, and this is a synaptic knob, and this may be our target organ or another neuron, but this synapsis right here occurs whenever the stimulation hits the end of this thing. We release something called a neurotransmitter. The neurotransmitter is released into this area and then is picked up by the target organ, whatever that target might be. And this is an actual picture of one occurring with an electron microscope. Synaptic function in neurotransmitters, acetylcholine, which is the primary neurotransmitter of the parasympathetic nervous system. Cholinergic synapses, the arrival of an action potential at the synaptic knob, the release of neurotransmitter acetylcholine, the binding of acetylcholine and the depolarization of the postsynaptic membrane, and the removal of acetylcholine by acetylcholinesterase. So we have axonal body, essentially target organ, axon. Information comes down to the end of this knob, and then we'll get a release of neurotransmitter into this area. The information is picked up on the opposite side, and whatever is left over, you get something called acetylcholinesterase that will essentially remove the leftover acetylcholine. And this is a picture of all of them together here. This is figure 811 in your book, cholinergic synapses. So information comes down. Exocytosis is how we release the neurotransmitter. The neurotransmitter on the postsynaptic neuron, which is here. Calcium is initiated, and then we get sodium channels propagating on this side, and another action potential occurs on the post side. Whatever's left over is consumed by acetylcholinesterase, which is those little Pac-Man looking things. So again, quick overview. Information comes down, neurotransmitter is released, neurotransmitter activates calcium, and then sodium channels open on the opposite side. Action potential occurs, acetylcholinesterase devours up the rest of the acetylcholine that's left over. Clinical note: organophosphate poisoning. And the organophosphate poisoning occurs from the deactivation of acetylcholinesterase, and this leads to an accumulation of acetylcholine. So if there's no little Pac-Man or no acetylcholinesterase to devour the acetylcholine, then we get an excess of it in this neuromuscular junction, or this synaptic knob, if you will, and this will cause sludge symptoms, muscarinic and nicotinic. And why those are caused is because the neurotransmitter that is there continually stimulates that site. Sludge stands for salivation, lacrimation, urination, defecation, gastrointestinal, and emesis. Urinary incontinence and bradycardia may also occur because of increased parasympathetic tone or vagal tone. On the nicotinic side, pallor, dilated pupils or mydriasis, hypertension, muscle fasciculations, which are like mini seizures, firing of motor end plates from the muscular viewpoint, cramps, and muscle weakness. Clinical note: neurotransmitters. 2 neurotransmitters of the autonomic nervous system. These are essential that you know these. One of them is acetylcholine. Preganglionic nerves of the sympathetic nervous system, pre and post-ganglionic nerves of the parasympathetic nervous system. Now, just to elaborate on this, whenever we're talking about the parasympathetic nervous system, it is on the pre and the post side that accepts acetylcholine. On the sympathetic nervous system, it may be initiated by acetylcholine. This would be sympathetic. But the post-receptor site may not be in this location. So generally what we see is this receptor site on this end, this ACH that's released from the sympathetic innervation activates a hormone or activates another body that will assist us in actually delivering the information to the target organ. An example of this is if you give someone a shot of adrenaline, you get a multitude of responses. Well, these multitude of responses are on receptor sites throughout the body. How we facilitate that is epinephrine is synthetic adrenaline, or epinephrine, norepinephrine are synthetic adrenalines. Norepinephrine is at the postganglionic site, the neurotransmitter of the sympathetic nervous system. Synapses that use acetylcholine both pre and post are considered cholinergic. Synapses that use norepinephrine as the end result and acetylcholine as the starting result are considered adrenergic. And you're going to need to know these terms. So if I have pre and post, and it is acetylcholine, then it's parasympathetic or cholinergic. If I have acetylcholine norepinephrine, it's sympathetic or adrenergic. Neuronal pools. Neuronal pool is a group of interconnected interneurons with specific functions. We have something called divergence. Information speeds from one neuron to several, and this is like speedy delivery. I'll show you in just a second. Sensory information. And then we have convergence, several neuron synapses on a single postsynaptic neuron, and this is voluntary and involuntary control that this will control here. So you have divergence. You essentially fire one somal body, and now you get a multitude responding. Or you have convergence. So this is receiving all this information and going to one somal body through dendrites. They would be at the synaptic terminal ends of these axonal bodies. This is the end of part 1. If you have any questions, please feel free to contact me, Roy Smith, 405-219-7613, or you can email me at smith@msa.net.